Welcome, paleopedologists. Today, I want to talk about the effects on soil formation and what we can learn from paleosols of early land plants. We're talking now Ordovician and Silurian age plants on the land. It's a remarkable event, the greening of the land, the coming of actual land plants. It could be, and it seems quite likely to me, that there were lichens, which is a microbial consortium, and also a variety of cyanobacteria and other creatures on land in the Precambrian. But the effects of plants themselves, the first non-vascular plants, have to have been pretty significant for the evolution of the world and its soils. Now, commonly, this topic is presented as the invasion of the land, as if there were armadas of algae and other sorts of plants that were trying to storm the beachhead, like the soldiers in Normandy, and establish a hold on the land itself. That's one way to look at it, I suppose, and it does seem true that land plants share a reproductive alternation of generations with seaweeds of various sorts. They also share plastids and other features that makes us think that seaweeds were their ancestors. However, it's also possible that land plants evolved on land, that land plants evolved from unicellular or palquicellular, just a few cells, a little green alga like Fritchiella, which has just a few cells, and that the various features of early land plants evolved in that lineage, not for the purpose of invasion, not for the purpose of driving off what was there before, but because it worked in an evolutionary sense. It could be that instead of an invasion of the land, we have a coevolution with the land by different sorts of soil algae, partly independent from what was going on in the ocean and in lakes at the same time. It could be that the evolution of early land plants, the early non-vascular land plants, was a case of learning, just like computers invented in the 1950s and filling a whole room are now compact enough to fit in our back pocket and do so many more things than we could have imagined. It could be that plants learned to live on land by natural selection, bestowing upon them a variety of features that enabled them to live in those sorts of environments. Now, it turns out here at the University of Oregon, we had a person who studied this problem extensively. Her name was Jane Gray, and she was a palynologist. She studied the kinds of organic remains that you can extract from rock by dissolving the rock in hydrofluoric acid. The idea was to see evidence of early land plants from their distinctive resistant features. She was most interested in spores, and one of her great discoveries was that in Ordovician rocks there were these rather remarkable spores, about 60 microns or so across. There were permanent tetrads called tetrahedroliths. Now, she knew that these were most likely the spores of liverworts. Because there are no algae that have spores with this kind of a resistant coat. This kind of a resistant sporopollen coat around the spores themselves enables them to be transported over land and to resist desiccation, um, attack by other organisms such as fungi, for example, and enables them to also be preserved in the rock. We don't get preserved green algal seaweed spores because they're soft-bodied and difficult to extract. This is a tough coating that she found in Ordovician age rocks. She also found that there were sheets of material. These thick cells that were an example of what's called a cuticle. A cuticle is a coating of waxy polymers of hydroxy fatty acids that is on the outside of all land plants. Now, algae don't need these kinds of waxy coats, kind of like an armorite coating that you might put in the back of your pickup, because they're in the water all the time. They don't dry out. But land plants always have them. So this was another clue that there was something going on here in terms of early land plant evolution in the Ordovician. And she also found tubes. These tubes appear to have been made of chitin or some rather tough material as well, like the cuticle itself. So this is 30 microns or so. These are the impressions of the cells. This also is about 20 microns or so in size. So Jane was one of the first to see from palynology, from bits and pieces that she was extracting from rocks, that there were land plants around in the middle Ordovician. I was very, very fortunate indeed just last year to be able to publish the first account of what these plants actually looked like. And this material came from an ancient karst deposit, an ancient cave deposit in Tennessee at a place called Douglas Dam. And it's the Douglas Lake member of the Lenoir limestone. And it's 470 million years before present. One of the fossils I found was this cluster of leaf-like things, and in the middle of them were these sporangia. The whole thing in side view would have looked like this, with the sporangia in the middle, these leaf-like things, and some rhizoids on the bottom. This one is Jane Greya. I actually named it after Jane Grey. The genus is Jane Greya. It's an example of a balloonwort. The modern species of that is Spirocarpus, which is found around Eugene. This is a non-vascular plant. It has no veins. It has no wood. It's an ephemeral or early successional plant, and that accounts for why Tetrahedralites is quite common in rocks of Ordovician age. The Douglas Lake member also had other kinds of plants as well. It had these rather interesting phalli with grooves and these prominent horns. Some of the horns were intact, others of them had split and curled. This is one I called Castellorum. And it's an example of a hornwort, another kind of a plant, which is a non-vascular plant that we find in moist areas today. It's actually most similar to Laosporoscis, which is a Panamanian hornwort. Another one had this dichotomizing thallus, and had these parasols of archegoniaphores, which were on this dichotomizing vellus. This is called cystites. This is an example of a liverwort. But my personal favorite from this assemblage was, of course, a plant which appears to have been a moss. It had a big sporangium, which was terminal, and then it had these rather wonderful frilly leaves on it. This one I call Dollyphyton, after Dolly Parton, of course, whose Dollywood is only a few miles from this locality. This is a kind of a sphagnum moss. It seems that in the Ordovician there were a variety of different sorts of plants around, early plants. That would have been quite a bit more sophisticated than just the microbial earths that existed during the late Precambrian. Now, Jane also studied the Silurian. And in Silurian age rocks, she found that the spores were suddenly starting to get rather more sophisticated. They had a trilete mark, or a hatching mark, a thin area of the exene where the spore could actually hatch. And they were getting these kinds of spines as well. This was pretty clearly a spore of a vascular plant, a plant that actually had tracheids. This kind of a spore is the sort of spore that is found in modern ferns, for example. This kind of spore is found in modern liverworts. This kind of spore is found in modern ferns. It's a pretty interesting kind of a spore because it's also found in some of the oldest vascular land plants that we know. This thing called Cooksonia, which is named after Isabel Cookson. These things are only about 2 centimeters tall, and they look for all the world like green matches. They had a terminal sporangia. They had a vascular strand down the middle. This is called the stele. And it has a particular kind of a tissue, which is called a tracheid. It's a kind of a tissue that makes up wood that has this biopolymer called lignin. The stele, there's a whole bunch of these tracheids right in the middle of the stem. This is the first, this Silurian plant, of the vascular land plants, but it's still herbaceous. It would have had about the texture of a bean sprout. This central vein or central stele of tracheids. And furthermore, she found that there were places where you could see on the cuticle, actually Jane and others found this, finally, some of these cuticles had what is a stomate, with a stomate. So instead of just having a flat-out cuticle with no openings, this had these water-retaining structures. So the cuticle goes like this. It thins over this area here, and thickens over the other cells. So here's the actual guard cells. These stomates can create an opening by pulling apart and becoming bean-shaped, or they can fluff up and they can close. And that means that CO2 can be taken into the plant for for creating photosynthesis, for creating organic matter, and oxygen and water can escape from the plant to keep it hydrated. It turns out that these tracheids of vascular plants were quite an important innovation in the evolution of plants, because they eventually enable plants to get big. The Cooksonia was already a little bit bigger than these tiny things. This is just about 4 millimeters tall. These probably only grew to about 3 inches or so. These were only about an inch. These only about a half an inch tall, because they didn't have that inner strengthening. So stomates, lignin, cuticle, spore coats, these are the features that make plants capable of living on land. Now, the remarkable thing about the Silurian, of course, is that it, with this diversifying set of inventions, vegetation was more capable of supporting animals. And there are a number of animals that came out on land, we think, quite early. Some of them we think were millipedes. Millipedes, of course, have 2 limbs for each segment. This is a Silurian millipede called Pneumodesmus. And tiny little spiders, like this one here, Paleopedology. When plants became more diverse, when they became vascular, we seem to have started to attract a burgeoning assemblage of animals. Now, the animal adaptations are distinctly terrestrial. These are generally burrowing creatures. These are capable of actually floating on the wind by trailing a little bit of web. They're so lightweight and small, just about 2 millimeters or so in size, but these have ancestors in marine forms like trilobites and eurypterids and others. They have an exoskeleton which gets them over the problem of dehydrating. They have limbs so they can move around equally on land as on the water. So what effect did these creatures have on paleosols? Well, we have a number of paleosols of Ordovician age, and I'll just talk about 2 of them. Some are in the Dunn Point Formation. Ordovician of Nova Scotia. These are quite extraordinary because they're between lava flows. The lava flows are columnar, so they were terrestrial lavas. They're not pillowed. They weren't deposited into a lake or into the sea. There's entrained soil material in the base of the lava flow, and the soils themselves are quite red. Red clay. There are reduction spots. And also calcareous nodules. So this is an A, BK profile. This is a C, and an R eventually. There's a sort of a zone of core stones here between the fresh basalt and the paleosol itself. This is a calcid, and a ridosol, which is, this is about 1 meter here, which is the kind of a soil that forms in an arid environment, a completely well-drained environment. The reduction spots are from burial glaciation of presumed plant organic matter, which was there during the burial of the soil itself, and was presumably some kind of a non-vascular land plant, like liverworts or hornworts, as we found in rocks a little bit older in the Ordovician in Tennessee. This is interesting because it's telling us that plants were not only in environments that were waterlogged and very humid, but also were in dry environments of well-drained soils at this time. This is not what we expect from modern liverworts. Of course, we don't find a lot of liverworts and non-vascular land plants out in deserts anymore because they wither and dry up. However, these plants are very poikilohydric. They can actually wither almost completely and not die out. They can refresh themselves, like resurrection plants, when conditions get better. These were actually studied by my student Lynn Fix. He did a lovely job on them. My personal favorites, however, and also studied by Lynn Fix, were plants in the Juniata Formation of Pennsylvania, particularly in an outcrop near Potter's Mills in Pennsylvania. These Juniata profiles were in sandstones, and what was remarkable about them was that they had these burrows. They were actually quite similar to the Dun Point ones. They were red, and they included calcareous nodules. So we have an A, a BK. That's kind of like a calcid paleosol. These are also Ordovician in age, late Ordovician in this particular case, very latest Ordovician. This is sandstone, and then there's more red beds down here with more burrows. This is just an AC profile, which we would classify as a fluvent. Once again, we have reduction spots that indicate that there was some vegetation here, all right, and these reduction spots would have been from the organic matter of creatures that were living in the soil and were buried with it. The burrows were really fantastic. And some people suggested that maybe these were marine burrows that were in the parent material and then the soil formed on that. I don't think so. And the reason is that we see in some cases the pedogenic carbonate crosses the burrow, and in other cases it is cut by the burrow. So this is carbonate, pedogenic carbonate, that was forming in the soil at the time when the burrows were forming and also after the burrows were formed. A really remarkable feature of the burrows were these backfill structures, which were kind of W-shaped. Backfills. Furthermore, some stretches of the burrows actually had fecal pellets in them. The burrows are about 1 centimeter in diameter. Actually, that was interesting, that the burrow diameter varied quite a bit and in a rather systematic fashion. So if you measured a lot of these burrows, I ended up measuring about 300 because there was some statistical problems with smaller counts. This is 1 and this is 2 centimeters in diameter. The distribution was like this. Striking individual spikes in the size distribution of these burrows. We know this kind of a size distribution, of course, is created by an arthropod. Arthropods are creatures that have an external skeleton, which is hard and durable. And so when they grow into that skeleton and until they outgrow it, at which point they shed it. And they shed the skeleton in a process called ecdysis, and they leave behind a molt. They'll often eat that molt because it's got precious phosphorus and other nutrients in it. But then they have to secrete another skeleton. They pump themselves up to a new height, and they secrete another skeleton. These are the molts of an arthropod that created these burrows in the soil. It had fecal pellets, which were actually coated with iron and organic matter, with a rather robust outer coating. That means that it probably was living in dry soil. This would have been a dry soil too, with a calcic horizon like so. And they were bilaterally symmetrical with this W shape, indicating 2 ranks of limbs in the soil. They weren't worm-like. They weren't creating a kind of a plug, a V-shaped or U-shaped plug. They were creating a W-shaped plug. So this means they were bilaterally symmetrical. I think they were created by millipedes. They were millipede burrows. I actually did name them. Squaenia biobaueri. And they remain almost the oldest evidence of millipedes in terrestrial environments. There's one older evidence. It's actually a trackway from the Lake District of Scotland, of England, which are just a little bit older. We have other fossil millipedes that are found in Cambrian marine rocks, and we're uncertain if the older millipedes lived on land or lived in the sea, where they are actually found. But it does look like from this soil that we have a situation where we had a creature that was living on land in a very dry climate, this very shallow calcic horizon rather than this rather deeper one. And some of the other tests that we ran on the chemistry of this soil indicated that it was rather cold as well, that the temperature was temperate in Pennsylvania at the time. Pennsylvania at that time would have been somewhat subtropical. So there is evidence for life on land in the Ordovician as well, from Paleopedology, and it seems to have been adapted already to rather unfavorable conditions. Another rather spectacular profile is found in the Need of Iron Formation. It's not a banded iron formation. This is at Iron Knob. Wisconsin, and it's also Ordovician in age. This one is on a very unusual kind of a parent material, and it's a kind of a soil that is covered up with dolostone. So it was covered by marine rocks, and it's a very, very thick, massive profile with a bit of clay in the top, and very, very, very red. This is now 3 meters. It's deeply, deeply weathered. It's very iron rich. It's basically a laterite. It has very low exchangeable bases. This would be a BW horizon. This goes down to a C, which is a sandstone, down through here. And study of isotopes in this profile are really quite interesting. If we look at the delta 13C of the material that's occluded in the goethite, now Creighton Yap did this work, we see that it has rather remarkably light values, -18, -14. This is in per mil or per thousand. This is a very light value, and it actually seems to reach an equilibrium value down through here. This suggests that this was a very productive soil, a rather productive soil in terms of its organic production of CO2, that it was respiring. The value of the atmosphere would be up here somewhere. And what happens in a soil with respired CO2 coming up, which is very isotopically light, is that it tends to offset the intrusion of air from the atmosphere, which is isotopically heavy. And the shape of this curve, for example, this would be a very unproductive soil, would have this shape. This indicates quite a bit of soil productivity. The mole fraction of carbonate too, included in this, also showed a kind of a bulge in productivity. This is 0.004, 0.0006. This particular paleosol is a kind of a tropical paleosol, which was quite productive. We don't know anything about its plants or its animals. It seems to have been a rather tropical soil. And using the Serling paleobarometer, we can calculate that it had about 300. It reflects an atmosphere with this amount of CO2 in the atmosphere. This was a time of a greenhouse spike. And this puzzled me for quite some time, because the other soil that I saw in Pennsylvania, which is late Ordovician, this is also late Ordovician, but not quite the latest Ordovician, indicated a rather low CO2 level of only about 300 ppm, and this one indicates a very high. This greenhouse spike is very interestingly timed. It belongs to a period of, if you look at the Ordovician, we've been able to reconstruct CO2 levels in the Ordovician. This is let's go down Cambrian here and up to Silurian here. We see CO2 levels were low. They were high in the Hernantian, also at other levels down into the Cambrian. This one here is the Hernantian. This is about 480 million years ago. This is about 430 million years ago. The Hernantian is a very, very interesting period of Earth history. It was the first of the really big mass extinctions. The mass extinction of the Hernantian was very debilitating for the Earth's fauna. All sorts of marine organisms went extinct. It's one of the big five, along with the Vranian-Furmanian or late Devonian extinction, the Permian-Triassic extinction, and the Cretaceous-Tertiary extinction. The Hernantian, though, is more famous for an ice age found primarily in the Sahara. In Mali, Mauritania, and Morocco, we have evidence of glaciers, glacial striations, tillites, a whole bunch of other glacial rocks that were very extensive on the African Craton at that time. This is the age of the Pennsylvanian Paleosols in the Juniata Formation. This is the age of the Nida Formation one. It looks like if we can get more of these Paleosols, we can reconstruct the history of atmospheric CO2 and the effects of land plants. This ice age was interrupted by the mass extinction. The mass extinction, we think, was probably due to a short-term massive injection of CO2 from flood lavas in Argentina. I visited that spot. There's a rather massive eruption of material which would have introduced a lot of CO2 into the atmosphere. But that CO2 was quickly taken up. And in fact, CO2 levels were relatively modest through most of the Ordovician, especially in the Paleosol in the Juniata Formation, because of the rise of the non-vascular land plants. It seems to me that the effect of land plants was generally a cooling effect. This ice age was inevitable. It was trending down toward that ice age. It was interrupted by big volcanic events, and there were also, at some of these earlier ones, there were quite massive asteroid impacts. But these temporary greenhouse spikes from transient perturbations were undone by the continued photosynthesis and soil formation on land. Tim Linton has made this case really quite beautifully, and I've stressed it myself. The idea is that plants cool by several mechanisms. One of them, of course, is straight up photosynthesis. Which gives reduced carbon. That pulls carbon out of the atmosphere. When plants become more complicated, like the early non-vascular land plants, we are accelerating photosynthesis in large areas of the land. That pulls carbon dioxide out of the atmosphere and has a chilling effect. It also gives the land vegetation a greater potential to recover from perturbations like this, from greenhouse spikes that cause mass extinction. But a more important way in which plants cool is called silicate weathering. So what happens in hydrolysis, of course, is we take feldspar plus HCO3, which is carbonic acid, to produce clay plus cations. And those cations are, of course, magnesium, calcium, sodium, and potassium. The feldspar itself is absorbing carbonic acid. It's taking carbon dioxide out of the atmosphere to create clay. It is also it is thus consuming carbonic acid with this particular soil reaction. When plants are cooling the environment, when plants are forming the soils, they accelerate weathering. They create more CO2 in the soil atmosphere, and that soil CO2 in the atmosphere weathers more feldspar to make more clay. It's a positive feedback system. Plants cool the environment. When plants have a major innovation in their biology and start to cover more land, they have a dramatic cooling effect. Animals, on the other hand, warm the environment. Animals don't photosynthesize. They don't take carbon dioxide in and create oxygen. They take in oxygen and create carbon dioxide. So we have this balance between the early land plants, the liverworts, the hornworts, the balloonworts, and the early animals, which would be the millipedes. We have this balance, which I like to call the Proserpina principle. The idea that this ice age, this Hernandian ice age, which was relatively short-lived, just a few million years, that this ice age was created by the evolution of plants, but shortly after the evolution of plants, of course, the animals went after them. These early non-vascular plants were delicious. Modern millipedes eat mainly debris. They do not do very well at all with woody material. In fact, the evolution of wood was probably an invention to deter millipedes. And the first woody plants, of course, appear up in here. Millipedes, however, do quite well in fruit and lettuce and other things that don't have a lot of these awful tracheids within them. The Proserpina principle, it could be that these events in the Ordovician created one of the great ice ages in Earth history, and also curbed a carbon dioxide greenhouse spike due to an early perturbation that created a mass extinction. So the evidence from paleosols is still ongoing. There are lots of paleosols to study, and we hope to learn more about these kinds of biological controls of Earth's climate through weathering and soil formation in future. And I'll go through some other examples next time when we talk about the even more dramatic but similar effect of the evolution of forests. And that'll do for today. Thanks.